Modeling cell division final assessment. The cell cycle. Interphase, G1, S, G2, cell division, mitosis, and cytokinesis.
Mitosis, the division of somatic cells. DNA can have two forms, chromatin or chromatid, an uncondensed form of DNA, or a chromosome, a condensed form of DNA. Mitosis contains 4 phases: prophase, metaphase, anaphase, telophase, and cytokinesis. Prophase is the first phase. Chromatin condense to become chromosome. Centrioles move to opposite sides of the cells and spindle fiber is created. The nuclear membrane starts to disappear. Metaphase, the second phase. Chromosomes line up along the center of the cell. Each centriole attached to spindle fibers which pull the chromosome to the center of the cell. Anaphase is the third phase. The sister chromatids separate and move to opposite sides of the cell. Telophase, phase 4. The spindle fibers disappear, chromosomes return to chromatin, and the nuclear membrane reforms. Cytokinesis happens when telophase does. It's the division of cytoplasm. It creates 2 daughter cells, and each daughter cell is identical to the parent cell. Meiosis is the division of sex cells. There's a mother cell and a father cell, and at the end of the division, it creates gametes, egg and sperm. Each mother and father cell have 46 chromosomes, but by the end, the egg and sperm only have 23. When the egg and sperm meet, they create a zygote. In meiosis, the cell divides twice, but its DNA is only replicated once. There's prophase, metaphase, anaphase, telophase, cytokinesis, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. In meiosis, genetic variation can occur. Crossing over and independent assortment are the 2 ways. Prophase 1 is when the chromatin condenses to form chromosome. The nuclear membrane disappears, and homologous chromosomes pair up to form a tetrad. Crossing over occurs here. Crossing over occurs to mix up the genes. This makes daughter cells that are not identical to the parent cell. Crossing over does not occur in mitosis because mitosis is the repairing of cells. Metaphase 1. The pairs of homologous chromosomes are arranged on the metaphase plate. Homologous chromosomes are split and move to opposite sides of the cell. Sister chromatids remain attached. Each half of the cell has a complete set of haploid chromosomes, but each chromosome is still composed of 2 sister chromatids. Cytokinesis, the cytoplasm splits and the chromosomes remain condensed. Prophase 2, the spindle fibers start to form and chromosomes move towards the middle of the cell. Metaphase 2, chromosomes are positioned on the metaphase plate. Crossing over caused the 2 sister chromatids to not genetically be identical. The spindle fibers attach to the centromere. Anaphase 2, sister chromatids are pulled to opposite sides of the cell. Telophase 2. The nuclei reforms and chromosomes begin to uncoil. Cytokinesis splits the cytoplasm and 4 new cells are created.